Hello, boys and girls. This is Mr. So. Welcome back for another lesson of biology. Today we're going through the chapter 26, Basic Genetics. Before you start the video, please remember to like and subscribe my channel, and also please share the video to your classmates to remind them to do their revision at home. Are you ready? Let's start. To begin with, if you want to study genetics, you must know DNA, which is the carrier of genetic information. So all our body characteristics, 我哋嘅特徵啦, they are all determined and controlled by genes, 基因. And what is genes? It is actually a part or a segment of DNA on the chromosome. Are you still confused? Don't worry, I will explain it to you step by step. Now the question is, do you still remember what is DNA and chromosome? I think this picture is very clear for you to understand what is DNA and chromosome. Here we go. So for the smallest part at the bottom here, which looks like a double helix, 雙螺旋, this is actually the DNA. And when the DNA, they try to coil up, 捲埋, and coil up some protein, and then they further coil up, and then coil up to something so big, it becomes the chromosome. So you should know DNA is very, very small, but when they coil up 捲埋, they become chromosome. DNA, it is just a short form. The exact name is deoxyribonucleic acid. Anyway, what you need to know is it does not have oxygen. No oxygen, deoxy, no oxygen, and we call it the short form DNA. This is our focus in this lesson. Right now, I'm going to go through a little bit of history of how the scientists discovered the DNA and their structure. Let's start with the year 1919. Scientists at that time, they know that DNA, they are the genetic materials, and they are made up of some very, very small molecules. And these very small molecules, they are the building block. They have a name. We give them the name called nucleotide. We break the words into different parts, nucleotides. And this nucleotides is made up of 3 different parts. Part number 1, we call it a 5-carbon sugar. This is a sugar, 糖. And then on the side next to it, this little ball is actually a phosphate group. And for the orange part in triangle, in the rectangle part, this one we call it a nitrogenous base. It is something related to nitrogen. For each of the building blocks here, for example, this is a single nucleotide. They are going to join with another nucleotide like this. They join with another nucleotide, and then they join with another nucleotide. So when they joined it together, we call this the very long chain in here, the polynucleotide. And when they joined it together, this part over here, you can actually see a backbone. Here we go, let's take a look. This backbone consists of a sugar, and then a phosphate group, and then another sugar, another phosphate group, another sugar, another phosphate group, another sugar, another phosphate group, and going on. So therefore, we call this backbone, which support the structure, the sugar-phosphate backbone. Scientists now they have a question. Does DNA exist in one chain only? Their answer is no, because for one of these polynucleotides, it is very unstable to exist on their own. Let's take a look. For the nucleotide here, each of them they have a nitrogenous base, and there are different types. They have A stand for adenine, T stand for thymine, C stand for cytosine, G stand for guanine. Each of them they are different nitrogenous base, and for the nitrogenous base, let me tell you, they are very, very unstable. So they cannot exist on their own. They must exist in pairs. The nitrogenous base must be pairing up with another nitrogenous base. Therefore, scientists they started to think DNA, it cannot be only one chain, it has to be 2 chain. And it might be a double helix, which you can see the base, join it up in here like this. So the base is going to face inward and join with another base of another chain. So now the scientists, they think, for the nitrogenous base, the ATCG, they must have a matching partner to join hand with. How do they join with each other? Does the A match with A, match with T, match with C, or match with G randomly? This scientist on the picture here, he actually finds out that the amount of A in the DNA is always equal to T, while for the amount of G is always equal to C. So this is his conclusion. Now, given that, they can now figure out the matching partner of the nitrogenous base. It should be A matching with T, G matching with C. Using the idea you just know that A always matches with T, and then C always matches with G, then here comes the question for year 2013, question 12. The DNA of a cell, eukaryotic cell, contains 20% of A, adenine. What is the percentage of cytosine based in this DNA? Alright, so let's think about it. If you have A equals to 20%, how many T do you have? T, it would also have 20%. And then what is the remaining percentage? Right now, you should have the remaining percentage of 60. So now the question is, what about C and G? Each of them, they should have 30%. So using the idea over here, so overall, you have 100%. So the amount and percentage of cytosine should be 30. The answer should be C. So after knowing that the A matches with T, C matches with G, you know that the double helix, they actually runs in two parallel chains, which have a matching partner. Let's take a look at the chain on the left. This side is the head, this side is the tail part. Alright, you can think about it in this way. And then for the chain on the right hand side, this part is the head, and this part is the tail. What do you see here? You see that these two chains in here, the left and the right, they actually have a opposite running direction. So we will say that they are running in opposite directions. And when the two parallel chains, after joining together, they wind up 捲埋啦. So you will see this is a double helix, 雙螺旋. After many years of hard work, when it goes to 1953, scientists, they finally have the conclusion of how DNA looks like. The two scientists, Watson and Crick, they proposed the model of Watson and Crick model. They conclude all the findings of scientists and said that A, it must pair up with T, C, it must pair up with G, and they are paired up using a very weak bonding. We call it the hydrogen bond. And we call this pairing A with T, C with G, give it a name, complementary base pair, a matching partner base pair. After knowing this structure and discovery of the DNA, what do you know? You know about the nature of science. 科學嘅本質啦. That means all science knowledge is actually tentative, 暫時性嘅啫. And subject to change. So scientists at the beginning, they don't know about DNA, but when the time goes on, they have more discovery, they have a better idea of DNA. And also, science is also affected by technology and the equipment available at that time. When times go on, you have better technology, you have better discovery. So you learned so much about DNA. What about another thing called RNA? It is related to the genetic materials as well. Let's take a look. For the DNA, their sugar is deoxyribose. That means no sugar. 模樣嘅。But for the RNA, they are called the ribose sugar. So actually, it is called 有氧. There is oxygen in the sugar. And one more thing is that for the DNA, it's actually double-stranded. It must exist in pairs. But for the RNA, it always exists on their own, single-stranded. If you take a look at the 5-carbon sugar of the DNA, it's actually deoxyribose. That means deoxy, no oxygen. But for the RNA, for the sugar, is actually ribose. That means it has oxygen. Okay, this is an oxygen. So for the final difference of DNA and RNA is that for DNA you have the nitrogenous base called a T, but for RNA, sorry, you don't have thymine, you don't have the letter T. The nitrogenous base T is changed to U, uracil. So this is another difference of DNA and RNA. Alright, that's it for the part of DNA and RNA. I hope to see you in the next lesson. Bye bye.